So a lot of things have changed in terms of the drug discovery pipeline. I mean, okay, so a lot of things have changed. After completion of Human Genome Project back in 2003, so many different targets became available. Earlier, it was more kind of serendipitous discovery or random screening, but a lot of things have changed back then. Since then, right? So now we have, the disease biology has become very important even before we synthesize the first molecule, where we take help from omics data, imaging, as well as the other data from different databases, right? So then comes the target identification. I told you, even though so many targets which you already know, but with the help of this very highly advanced data, we can always prioritize the target, identify and validate these targets. So target ranking is crucial, where AI is helping. And at the next case, that is the virtual screen, where AI can now handle chemical space docking, as in 10 raised to 17 molecules in one go, billions, trillions, if not thousands of trillions for that matter, zillions of molecules. So it is now possible. So AI has definitely made a mark when it comes to virtual screening and chemical space docking, because now we have so many different possibilities, including the molecules which are generated by AI, and everything is just happening at such a lightning speed, right? Very high speed in no time for that matter. Then comes lead optimization. Now again, when it comes to lead optimization, you have so many properties to balance from. This is where AI can take a holistic view and help us optimize the lead to move ahead where it comes to SAR, that is structure activity relationship, and all the admin data, absorption, distribution, metabolism, excretion, as well as toxicity for that matter. Can I request the participants to switch off their mics, switch off their, put them on the mute so that it doesn't disturb me. Otherwise, these disturbances will actually break my link and I won't be able to do the job I'm supposed to do today, right? So it's a sincere request. Please do so. Then comes preclinical profiling, where you have the toxicity as well as pharmacokinetics risk. So there also, as you go from hit finding to lead optimization to preclinical to clinical, things become very, 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 very complex. And this is where AI can potentially help for multiple things. It's not just one particular aspect, but multiple aspects. And this is where the last step being the clinical translation. Can I ask the organizers to switch everybody? Otherwise, I'm just going to stop and leave the meeting. I cannot really tolerate this. Sir, can I ask you to switch off everybody or remove those participants who are creating the nuisance? Otherwise, I'll just log out of the meeting and stop the lecture here. I'm spending my time, very important time, just so everybody benefits. You have to respect my time, please.
We request all the participants to kindly mute yourself. We will send message to everyone, sir.
Okay, ma'am. Thank you. Okay, so having that, so then in the again clinical translation, there is so much that we need to do, because there is it's one of the most complex areas of the complete drug discovery and development pipeline, right? So trial enrichment, so where you actually increase the probability of a successful clinical trial outcome. Because ultimately, when you file the new drug application NDA, you have to tell the FDA or the regulatory agency that the benefits outweigh the risk. And this is where the clinical translation, where AI can play a key role, right? So we talk about that. So the key principle is every prediction must eventually connect to a measurable decision. Just say pointer option. To a measurable decision, assay selection, which includes assay selection, compound synthesis, dose choice, or patient stratification from the clinical trial perspective for that matter. Right. So having said that, let us come back to the vocabulary of AI. which includes AI, ML, that is artificial intelligence, machine learning, deep learning, generative AI, right? So now you see there is a layered approach. At the top of it, or the biggest area or the general area is artificial intelligence, where machine learning is a subset, deep learning is a subset of machine learning, and generative AI is the subset of deep learning. So you see there are multiple layers. When I say artificial intelligence, so I'm talking about engineering of machines and programs that can learn, reason, and make smart decisions like humans, right? So this contains everything. That's the superset, I would say, artificial intelligence. Machine learning being the subset of AI or artificial intelligence, machine learning is nothing but science of teaching, a science of teaching computers to learn from data without being explicitly programmed. Now, this is another very important function. So you have a machine learning model. You're not explicitly programmed, so but then the model will learn from the data without being explicitly programmed. Then comes deep learning, which typically involves the neural networks, artificial neural network, or ANN. Again, this is a subset of machine learning, which is so deep learning, or DL, is ML using deep neural networks that learn complex patterns on their own. So if you look at the architecture of any neural network, so you have the input layer, you have the output layer. So data goes in the hidden layer. There are multiple layers for that matter. And here, what happens in the hidden layer, nobody knows. But then whatever happens, it gives us the expected output. And this is exactly where the deep learning has a unique place in the overall architecture, including machine learning and artificial intelligence. Then the latest technology or latest terminology, I would say, is generative AI, which is nothing but AI that produces new content such as text, code, images, molecules, summaries, questions, and hypotheses. So in a way, if you look at the LLMs for that matter, they can potentially be considered as a generative AI, where you give a particular prompt and that AI model will work on that at different levels depending on what question you have asked. Then it will look for appropriate data and it will generate the new content. It could also generate summary, molecule, and multiple other things, right? So I'm sure everybody is using Chat GPT or Claude or Gemini or other tools for that matter. Right. So then that brings us to another question, that is like, what is an AI model? Right. So because we talked about what is an AI, what is ML, what is DL, what is generative AI, but then the fundamental operative is the AI model. Right. So what is an AI model? Right. So now you can see here again, the workflow is given. An AI model essentially is a culmination of multiple things where you actually input the compound, assay, protein, or any text. So you need an input. So the moment you say input, so you are inputting, say, a compound. So then the compound is represented in multiple formats. Could be smile string, could be 3D confirmation, could be fingerprint, multiple representation. Because now you see, you're talking to a machine, you're talking to an AI. Now, AI is not, that machine is not human, it's a machine, right? So the way you talk to another human, the way the human will understand, the AI will not understand, the computer will not understand. So it needs a different way of understanding things. I mean, if you just search how AI processes a simple query when you put it in Chat GPT or Claude, so it's very complex actually. Everything happens in real time, and that's where when you get the response, so you see that it is appearing word by word, right? Because the model behind it is working fully to generate what you have asked for. It has to make connections, it has to extract information, it has to present that particular data the way you are expecting, right? So a lot of things happen, and then the next part is the model learned parameter. So depending on, like I said, today's AI models, they are multimodal, meaning the data itself is multimodal. So it has to understand an image, it has to understand the number, it has to understand the biological context, it has to understand the pathway, it has to understand the mechanism. So to do so, the model will learn some parameters to answer a particular question. It could be, like I said, Chat GPT, or it could be a model which is now from the chemical structure. It is predicting the aqueous solubility for the given molecule, right? So the model, in the background, without you even learning or understanding, the model will generate these parameters from, of course, the model has been trained on a large training data, and then the model will just try and use this learned parameters for giving you the appropriate output, which could be a score, like output score and uncertainty. Like the model will tell you that, okay, it has given out some output, but whether this output is reliable, whether his output is credible, right? So that also the model will test, so that the model has generated something, and you will use these results. You will use these results with a pinch of salt, meaning whether this is uncertain or the model is very confident, right? So we'll again talk about that thing in some time for that matter. Right, so here, for example, given this smile string, estimate aqueous solubility and confidence. In fact, we will be doing that in some time. So you give any smile string, I'm sure you understand what smiles is, right? So it is a 1D representation of a chemical simplified molecular input line entry system. That is smile string. Right, so you give the smile string, which is 1D representation, and generate some number, which is say the predicted aqueous solubility and the confidence with which it predicts, right? So it will also tell that it is say 10% sure that the aqueous solubility is likely to be this. And in other case, it may say for other molecule that it is 90% sure. So obviously, I will believe the one which it is saying that it is 90% sure, because the model has been trained with that kind of data where the chemistry of the query molecule matches in a way partly or fully with the chemistry of the training data. So then it becomes more confident in predicting what that particular thing is, and this is exactly where that confidence is very important, and everybody should always be sure to understand what the model is trying to tell you or how the model has evolved, because otherwise it will just generate some number, and if you blindly follow these numbers, it's really going to make life difficult for you, right? So you have to understand the model is not magic. It is not like you put something in the box and the magician turns it into something else which you think is not possible. So it's not magic. It learns the statistical relationship from examples and usually fails outside its experience, meaning whatever data it is trained at, it will do things extremely well. But the moment it encounters a chemistry out of the training data, it will not do so well. But it will tell you, it is up to you whether to blindly believe or just ignore the warning that the model is giving you. Right, so that brings us to the very sense of AI modeling or AI, the food, the raw thing that the AI model needs. Right, so this drug discovery data, it's multimodal by nature. That means different types of data are seen. Meaning, like I said, there are chemical structures where you represent the chemical structure as a smile string 1D data, or it could be 2D SDF, that is structure description file, or 3D SDF. Then there are scaffolds, meaning the core structures or substructures of the molecule, common substructures, and as well as fingerprints. So you see, there are multiple ways for representing simple chemical structures. Then comes the bioactivity. Bioactivity is again in different forms. It could be IC50, it could be EC50, it could be the inhibition constant, it could be the phenotypic readout, like change in the size of the adipocyte upon treatment with the drug for that matter. Then
ఫస్ట్ పేరు ఇంకేంటి పర్మిట్
ప్రశాంత్ సర్కార్ ఫస్ట్ నేమ్.
hello,
Hello, sir.
am I audible?
Yes, sir. Yes, sir.
Okay. All right. So Okay, so then the data coming from biology or omics data, very complex data coming from genomics, proteomics, the pathways, the disease signatures, so many data. It could be numerical data, it could be images, it could be some patterns, it could be multiple things. Then comes the structures or images. It could be a PDB 3D structure, it could be cryo-EM structure, it could be microscopic structure. It's like the pathology that you essentially take it for say cancerous tissue. So the data is coming from microscopy, pathology, for that matter. At the same time, the data can also be in the form of a text or knowledge, which is derived from multiple sources, including patents, papers, protocols, electronic lab notebooks like that. And of course, clinical or real world data, where the data includes the electronic health records, biomarkers, and trial outcome. You see, there is so much variety of data. It's not just the variety, it's also the nature as well as the humongous amount that the data is available in, right? So this is where so then that brings us to the next question. Okay, I draw a structure, my students will understand this is some structure, paracetamol, this, that, whatever. So, but then machines wouldn't know what this is. So machines, they have to see the molecules, they have to understand. So how machines will see the molecules? So in case of smile string, so this is just a smile string for one particular molecule. Now here, I think this is paracetamol. Yeah, it is paracetamol. Okay, so there is a smile string, which is a 1D representation of the structure, which is a compact string, easy for language models. So the large language models understand it very well. Then comes the descriptor. So where you are representing a structure in the form of its molecular weight, log P, topological polar surface area, hydrogen bond donor, acceptor numbers, and so many different, maybe more than 5,000 different descriptors. So it's a human interpretable figures or features, I would say. Then comes fingerprint. So usually a binary fingerprint, which will be a unique combination of digits based on what information it is trying to trap, 01000 like that, zeros and ones. So it's a substructure presence or absence. It will just try and look for that. Then comes graphs, where you are represent molecular graph. Basically, each molecule is treated as a graph where atoms are nodes and bonds and as edges. So you are connecting the two nodes by a bond by as an edge. So the unique representation that comes out of every molecule, the AI will learn from that. And when you are trying to look at patterns, say with reference to biological activity, definitely will give you that particular edge for understanding how the machines are seeing these molecules, and it can process because of the humongous computation power. It can see a lot. It can learn in very small amount of time the native chemistry topology, which can be useful for multiple applications like retrosynthesis. Then, of course, the 3D conformations, how the molecule behaves in the 3D space or 3D structure space, where you have got the XYZ coordinates and the electrostatic features which are trapped here. These are usually useful for structure-based modeling, where we are trying to dock a molecule in the active site. So how the shape complementarity and the electrostatic complementarity, for that you definitely need the 3D conformer. So depending on the context, depending on the question that you ask, the machines will see the molecules and process the information accordingly. So on the other side, how do machines see the targets and biology? Now, this is again very important, because without targets, there are no drugs. And this is exactly where how do machines see the targets, and how do they try to find the patterns, how, like, what molecules are likely to bind to a binding site in the target molecule. When I say target, I'm talking about the 3D macromolecule. So you have amino acid sequence, which is again a 1D representation. So protein language models, homology, and domain analysis, they will use the amino acid sequence, trying to understand what part of amino acid sequence is common to say 10 different totally unrelated proteins, and is there anything related to the 3D structure. So that brings us to the discussion on 3D structure, where you are more interested in how the binding site shape is, so that I can design molecules which will occupy this binding site, adopt that particular shape, and show activity from that binding for that matter. So these would essentially be used for docking, pocket, similarity, interaction, prediction. Then comes expression signature. So when a particular molecule, a patient is treated with a particular molecule, what is the expression signature? And I'm sure you know, suppose somebody is detected, some female is detected of say breast cancer. The first thing that they will do is they will actually take out a small piece of the cancerous tissue biopsy, and then they'll figure out what type of, like, what is the genetic signature for that. So whether it is ER positive, PR positive, ERBB positive, or it is triple negative like that, because based on that genetic signature, essentially, you can potentially decide which drugs to use for the therapy. So likewise, the expression signatures based on the treatment, following the treatment of the molecule, say for a novel molecule, newer molecule, which is under development, how exactly the, like, which genes are being expressed, which genes are going down as far as their expression pattern is concerned, because which can unravel the importance, or I would say important insights on the target discovery, as well as finding the new uses of the already existing drug, that is drug repurposing. Right. So here, the next level is the pathway graph, where you are looking at the pathways. Where you're looking at the pathways as nodes, and like what is the upstream, what are the upstream molecules, what is the product, like how the pathway is progressing, how the pathway is affecting. So the nodes and edges, which are useful for knowledge graphs as well as causal hypothesis when you talk about the mechanistic aspects. Then comes images, which is about the cell morphology or histology. Following the drug treatment, how the cell is changing, or how the histology or disease pathology is changing. So here you're looking, or rather looking at the phenotypic screening or digital biomarkers, right? So how the machines will see targets and biology. Then comes the next important thing, that is the algorithm. Now, data quality beats algorithm. That means if your data is of poor quality, even if you have access to the best algorithms, they will not really do well when it comes to model training, right? So most AI failure in discovery starts before the model training. That means unless you are very careful about the data that you are going to use for model training, otherwise you are going to generate some models which are good for nothing, futile models. Now, in this particular case, say acid drift. Now, some biological data is complex, very high variability. So in that case, if you take the data, say for anti-cancer activity, different types of data. So every assay will have a different drift or different uniqueness. So if you use data from different protocols, batches, or readouts, in that case, the data is not homogeneous. It is likely your models are going to be all futile. Then label noise. So in this case, the IC50 values depend on assay condition. So for the same molecule, you will see 10 to 20 different biological activities or IC50 values are reported, but then which one is that you will take for model development? So that is, because most of it will be label. I mean, it will be a label noise. Then comes the imbalance, where you're talking about use of the imbalance in the selection of data. I mean, there are too many inactive molecules and few active molecules. So the model will not be able to differentiate between the activity and inactivity, and it will not be able to do justice. So these are some of the cautions or care should be taken for addressing these before even you start the model training. Then comes data leakage. So you are trying to use very similar molecules, in the training set as well as in the test set. So the data which is in the training set will be used for model training, but at the same time, the test set which is not used for model training or model development will be used for making sure that the model is doing well. So this is where the data leakage, that's another thing. So that is one thing. Then missing negatives. So otherwise, if you use too many if there are no negatives, the model will not be able to learn. So here, this is a very important statement. Absence of evidence does not necessarily be equal to evidence of absence, meaning some molecule, now you see, just to give you an example, many molecules when they are used in cell culture assays, like in the cell-based assays, so many molecules do not show activity. But then, when you go to the higher level, they usually show activity. Many anti-cancer drugs do not show any activity in the cell-based model. Why so? Because suppose if the target is inside the cell and the molecule is not able to get inside the cell, so the cell-based assay is not really going to show any activity, right? So in that case, this is where I'm saying absence of evidence that is not necessarily equal to evidence of absence. This is very important, meaning a molecule is not active because it is not active, or the molecule is not active because it is not able to get inside the inside that particular cell, right? So this is important. Then comes the model training workflow. So you have taken care, necessary care as far as the data is concerned. Now the next thing is how you train your model. So model training workflow. So first you have to define the end point. What decision will be supported? So are you trying to estimate the aqueous solubility, permeability, or you're trying to understand what is going to be the clearance or metabolic liability, all all of this, or half-life, intrinsic clearance, whatever it is. What decision will be supported? Because this is very important part which you define before you even collect the data and curate the data. So then you have, when I say curating the data, suppose you go to Campbell and then try and extract data for one particular say kinase inhibition data. So in that case, you will see data in the form of Ki, you will see data in the form of IC50. So you have to make sure that you remove the duplicates, you remove the additional things, and then you also make sure you use the correct unit because IC50 in say nanomolar versus Ki in nanomolar, these are two different units, two different things, or even percent inhibition at one particular concentration. So you need to curate the data so that you do justice with the model development, meaning you feed the correct data to the to that particular model. Then only the model will come out to be good and give you what you are expecting it to do or the job. Then the next thing is you need to split the data, meaning you have to suppose if you have got 10,000 observations, you need to maybe 80,000 observations will go training data and 20,000 observations will go in the say test data, because or it could be training, validation, and the test data. So training data will be used for training the model, validation data will be used for validating or selecting the best model and validating it, and the test data essentially will be used for testing the robustness of the model or how good the model is. So that is one. Splitting the data includes this, avoid the leakage or scaffold split. Then the actual process of training the model, where you are looking at the baseline model and then you are looking at the complex model. So when I say baseline model, the minimum or the model containing the minimum number of parameters or features, the most complex will be the model containing all the parameters for that matter. So this is where model development itself or training the model or model training itself is a complex process. So there are multiple matrices that you are going to use for making sure that you come out with a lesser complex but yet best model or rather better model. The next thing is definitely the internal validation and the external validation and the prospective validation, meaning when you are going ahead, you are actually validating the model while the model is still being used for during its active life. When I say internal validation, so use the training data and delete one molecule at a time and then try and use the model for, meaning develop the model for remaining molecules and predict the activity of the molecule which is omitted, say leave one out, cross validation for that matter. So likewise, there are multiple ways and multiple matrices which essentially can be used. And then the last step is the deployment of the model. And then that's where you will monitor the drift and update it carefully. Right. So it's not that your job is not over once you develop the model, and do not worry about how the user is using or what the user experiences. So there are continuous improvements that will happen as and when the new data is available, as and when some issue arises, in the model which you have developed. Right. So model is only as trustworthy as the question that you are asking it to. You remember the first one, defining the endpoint data, how good the data is, split strategy, and of course the external validation behind it. So you have to actually challenge the model. Look, this is what the data was used, training data was used, but now you have a data which you have not seen. If you are really good, show prove your worth. How this particular model is. meaning how good you are in terms of prediction. In fact, there are a lot of blind challenges, open challenges that happen. So where the data, training data will be made available to the users, and then there will be a test set for which the actual data or experimental data is masked or blinded. And then if you are good enough, you develop the model and try and do well with this particular data. That is the test data, right? So if your model is really good, so you need to prove your worth, and this is extremely important. Then this is what I was talking about, the validation matrices. So you have to choose what matches the decision, right? So yeah, so here for regression modeling, you are using the root mean square error or then the R square value. This is typically for property prediction. In case of classification model, binary classification, yes, no, 0, 1, black, white. So you are using the ROC AUC, receptor operating curve, receiver operating curve, and the area under the curve. And then there are multiple other matrices: sensitivity, specificity, false positives, false negative. So typically for activity or toxicity labels, whether the model is meaning whether this molecule is going to be active or inactive, toxic, non-toxic, that kind of thing. If you are ranking, so these are different types of models. So if it's for ranking, so you are using the enrichment factor, hit rate, and top K, this is precision. In the sense, suppose if you are working with a particular say docking algorithm, right? So in that case, you will also add the known molecules, some known actives, and then if the model is really good, the model should enrich those active molecules in the top, say top 1%, top 5%. So that is what is the enrichment factor, meaning it should be able to rank the molecules. If it is able to do, if it is able to rank the molecules, meaning the known actives, then definitely it is good. In that case, whatever molecules it has ranked, they have a very high probability that it is going to be high ranking, or rather the model is going to be good. And then of course the calibration, so confidence versus actual error. So you need to understand how the model is predicting and how these values are essentially close to the experimental values, the difference, or I would say the deviation between the observed and the actual one. So that is definitely give you an idea as to how the model is good. So this is where it will also tell you the decision risk, whether the risk will be higher or risk will be lower. You typically would want lower risk. Right. Then comes the applicability, domain, and uncertainty. Like I said, the model, if the model has the training chemistry, meaning the query chemistry is very close to the training, meaning say you give a particular smile string for aqueous solubility prediction. So if this chemistry of your query molecule is very different than the molecules on which the model is trained, so then it is definitely not going to do well. If it is close, then in that case, the predictions will be very high, the risk will be lower. Regarding the project chemistry, you need to be very cautious because the results will be like 50-50. So you need to take your judgment. And if it is a completely novel space, then there will be a very high uncertainty. And this is why we need these models to get trained again and again and again. And in the sense, updated, improved as far as their prediction is concerned. Right, so it's very important. So that's the only thing is you can actually keep on adding the chemistry space, and then that is going to be very helpful. Then, so now we are going to talk about the utility of the AI/ML models in multiple areas along the drug discovery pipeline, where say for that matter, target identification and validation. So AI can help connecting disease signals to actionable biological hypothesis, meaning you see that there is a particular phenotype in a particular molecule. And then if there could be multiple hundreds of thousands of reasons why you see so, particularly this happens in cancer patient because it's a very, very heterogeneous disease. In that case, you can potentially get the data from omics, literature, genetics, like I told you about the breast cancer patient, what is the genetic signature. And then all of this data can be put together for integrated target ranking and evidence map. So you have data from the clinic, some observation, and you have data from the omics, from the literature, and you're just trying to connect the dots just to understand what is the underlying pathology behind it, which pathway is this regulated, and what is the right way to disrupt this pathway. I mean, there are still so many different diseases for which there is no disease, diseases and disorders for which there is no drug, right? And this is where you understand, you try to understand the pathways. Then you also take the human review and then look at the mechanism plus tractability, meaning whether this target is going to be really useful or there is something else that needs to be looked at. So basically, AI can help you in target identification. So this is the activity that goes on in big pharma companies like Merck, Pfizer, so continuously, because they have access to all the crucial data from very highly advanced technology, something known as spatial transcriptomics, like that, which nobody has access to. I mean, very few will have access to. And here you are also trying to look at the phenotypes, where all of this will lead to target identification, and then ultimately experiments can be planned for validation, right? So the critical question is the target causal, meaning there is indeed a disruption in the target function and the disease. There is a cause and effect relationship, whether the target is druggable, meaning you have identified the target, but there is no way, it's a protein-protein interaction. There is no way you can actually, the target is druggable or assayable. So you have to actually develop an assay to identify molecules which are going to be active against that target, and then of course safe enough to justify a program. So again, all of these questions are asked during the early phases, that is target identification and validation, without which you won't really initiate a program if the target is a surrogate target or there is an alternate pathway involved. Even if you inhibit the target, whether that essentially is going to there will be some other turnaround. In that case, the inhibition of target will not really be any use of any use, and this is where you need to look at it. Okay, the next use case of AI is the protein structure prediction. Now, I'm sure everybody knows about AlphaFold. A couple of years ago, or 3 years ago, three guys from the protein structure prediction and protein design were awarded Nobel Prize. Like John Jumper is one of them, and then there is David Baker for protein designing, and then John Jumper's boss. So they got John Jumper and his boss got the Nobel Prize for protein structure prediction using AlphaFold. Google DeepMind, that's a company, right? So because see, like I said, if you look at the 3D structures in PDB, you have say 250,000 structures available of multiple different proteins. There are so many proteins for which we don't have structures available. But at the same time, you have cryo-electron microscopy, which essentially is able to give structures of very, very complex proteins, multi-domain proteins. So why do we need these 3D structures? We need these 3D structures to understand, of course, the design aspect, to understand how the molecules, small molecules, macromolecules, biologics are going to interact. Right, so protein structure prediction. So the AI, so AlphaFold has used multiple AI models to predict the 3D structures only from sequence. So you may design a completely novel protein and still be able to, have the AI predicted 3D structure, which can help you to do multiple things. And this is what AlphaFold has done. Of course, there are multiple other definitely good for predicting the 3D structure, right? So what changed when AlphaFold came into existence? So now we have large scale access to predicted protein structure, which I mean, I can just go to AlphaFold, look for any protein for which there is no experimental 3D structure available. I can just take this particular structure and start my virtual screening right away. Better starting points for target assessment and mutational analysis. So because when it comes to sequence, the sequence will always have meaning. Basically, the structure function analysis, you change the structure or you change the sequence and the function will be changed, right? This is where you have now better starting points for. So again, when I say mutational analysis, depending on what I see in the structure, I can potentially mutate and see what effect these mutations will have on the overall 3D structure stability. And then, of course, new opportunities for biology education and structural reasoning. I mean, availability of 3D structures of macromolecules is central to biology, again, from the perspective of structure function analysis. There are so many different things of proteins and other macromolecules which we don't know the structure. We don't know why they exist in the first place. And that's where the 3D structures are going to change the whole thing. Then what remains difficult despite the AlphaFold being there? So we still are struggling with multi-domain proteins, the conformation, disorder, and these are all the proteins like apoprotein structures, or there is no cofactor, no ligand, water networks or induced fit. So that is one. Then ligandability and true binding energetics, because there is no ligand, it becomes difficult to visualize where the binding site will be, or even if you have some idea about the binding site, using these structures becomes very, very difficult. And then, of course, assay relevance and experimental confirmation, despite the availability of newer 3D structures or proteins, could be new protein, like I said, we don't know about the assay relevance and experimental confirmation. That still remains difficult. So that brings us to the next important things based on next important aspect of drug discovery, that is structure-based AI, which uses the 3D structure of the protein to be able to do what it is supposed to do, that is for designing new molecules. And this is where, excuse me. So this is where the structure-based AI comes in picture, where you're using it for docking, scoring, as well as rescoring. So AI often improves the ranking, but physics and medicinal chemistry remain central. So you get from the AI predicted 3D structure, you identify the protein pocket, you take the compound library, could be completely AI generated or molecular generated library, and then you do the docking and poses, and then finally you have to again look at from the physics-based docking, like docking programs and all. Right, so AI or physics-based rescoring, and then you test the topics, could be novel molecules. Just one particular figure from my own project is shown. So there is a tetrapeptide which we actually designed these tetrapeptides and then did the docking, and then this is how it is interacting. So part of this work was done by the AI, including the screening of, like, virtual screening of the design peptides. We did the designing peptides, so designing of peptide. So then, of course, the next important activity during the drug discovery pipeline is the lead optimization, where I said it's a multi-objective optimization, multiple parameters you have to optimize, and usually you try to change one, the other one gets derailed. So this is like walking on a rope, and deep down on both sides there is a deep valley. So if you do not balance well, you are gone. You will go in the valley, that you will fall in the valley. So potency is very important, potency of the molecule and selectivity for the given target. Apart from that, you have to work around solubility, because unless the compound is soluble, it's not going to do anything. If it is not permeable, it will not get inside the body. If it gets degraded, like metabolized, so metabolic stability is important. It will not be able to reach its site of action. Safety, if it creates a lot of other issues of target effect, then that is a problem. And then even if you design a molecule which is having everything, but if it is not able, if you are not able to synthesize it, it is good for nothing. And then at the same time, even if it is synthesizable, but if it is not patentable, there is no fun in doing anything. And this is exactly where, so you have to just see how many properties you have to balance, and this is where the AI can help in balancing potency, selectivity, adamant, IP, and synthesis aspects. So all objectives must be considered together. So optimizing of one often worsens another. Then, of course, like I said, nowadays everybody talks about AI-assisted retrosynthesis and synthetic feasibility. Of course, just by looking at the structure, the AI will tell you how easy or difficult that this molecule is for synthesizing. I mean, we do have access to, say, SciFinder. Whatever chemistry is known so far, everything is there. But chemistry is so vast, whatever molecules that have been reported, despite the number runs in few billions, but that's not all of the chemical universe. You may still Okay. And that's what typically happens in drug discovery. So then how to synthesize this molecule? I mean, there is no literature precedence, and this is exactly where you come back to AI-assisted retrosynthesis, where the AI has learned the chemistry. The moment you give any molecule, it will tell you how to synthesize this molecule. But then here you have to use your human judgment to make sure that the molecule is, meaning the retrosynthesis pathway is feasible. It's not just possible, it's feasible in the sense you can make it in the lab. And this is where the AI models are proving out to be very, very good in terms of AI-assisted retrosynthesis. So AI can suggest synthetic routes, synthetic chemists decide what is robust, safe, scalable, and publishable. The human in the loop connection. Then comes the knowledge graphs and literature mining. Now, of course, everybody is using Chat GPT and this and that, and this is exactly where the AI can help. So here, when you talk about drug discovery and the knowledge that is generated along the way, so drug discovery is full of hidden relationships that AI can help resurface. So because AI can look at multimodal data and human mind at times cannot really perceive in timely manner. Of course, everything is possible. Humans can do it, but it is going to take time. And this is where the AI can go deep down, take the deep dive in the disease biology, target biology, pathway analysis, compound properties, phenotype, and trial, and everything. It can find these hidden relationships. That is extremely cool, right? So this relationships could be in the form of evidence, some evidence that we are not aware of, activation, inhibition, association, or trial outcome, right? So it extracts the knowledge graphs. They extract entities from papers, patents, reports, and ELNs. It is electronic lab notebooks, which is actually a proprietary data, and it's like humongous data. Then connect compounds with genes, targets, phenotypes, disease, and assays, and at the same time support the repurposing. It is finding new uses of old drugs or approved drugs, target assessment, and competitive intelligence. So much of this, right? So then comes large language models in drug discovery. Now, every other day you see that Chat GPT has come up with a unique model. Cloud has come up with cloud science. There is so much that is actually being done. So these large language models are really going to be extremely useful. So these, when I say large language models, most of these are generic models, but then now they are being specifically developed for chemistry, for drug discovery. So you give a query and the model, because it has learned on a very large data, a specific data for that particular domain or field, it is able to now give you very, very fundamental, very, very fantastic outcome. So LLMs are useful interfaces and reasoning assistants, but they must be grounded in the sense, a lot of times these models hallucinate and it generates data which do not exist. So you should be very careful, and this is where human in the loop connection comes in picture. Then comes, like, if you look at the roles, useful roles, so summarize the literature and protocols, draft code or analysis workflows, query internal knowledge bases, which could be a proprietary data, generate hypotheses and experimental plans. The known risks are hallucinated citations or mechanisms. If you look at the statistics, more than 100,000 or 125,000 false or hallucinated references have, because the users were not careful, meaning the papers exist, they have been published, but then they contain citations which do not exist, generated by AI. That's very, very potential risk. Confident but unsupported claim. So it will tell you that I'm confident, but that there is no support. That's the claim that it is trying to make. Confidentiality and IP leakage. This is very important. So whenever you are putting any data on these LLMs, all of these data can be seen by their owners. They can use it potentially. That's where you should be careful about putting confidential data for the general purpose LLMs. Weak chemistry without tools and data. That's where the LLMs, unless they're trained on large amount of chemistry data, their predictions are going to be weak. So the practical use is use LLMs for drafting and synthesis of evidence, use validated cheminformatics and experimental data for decisions, right? So again, when I say cheminformatics, again, most of what we are discussing as far as, say, related to the structures, that comes under cheminformatics, right? So, and then of course experimental data for decision. So AI integrated, AI enabled hit to lead workflow, which is the recent one. So you start with so the strongest workflows combine the prediction, design, synthesis, and experimental feedback. It's like you execute the workflow, you generate the experimental data, and then again you pump in this data in the workflow so that the model is trained better. So it's like you have a model for accurate solubility prediction, and you so it is giving you some output, and then based on that you test the experimental, you do the experimental data. Some of the data matches, again you can still give the data and update the model, or even if it does not matches, you can tell the model, and then eventually the model will recalibrate itself and then give you amazing data. So again, as and when the new data is available, you should actually again go and retrain your model. So this is always a continuous process till the model does not improve. Then here case study one, AlphaFold as a teaching gateway. Now here, in this particular case, you can actually choose a particular target, any target that either you are working or about that target. Then you inspect the confidence. Basically, go to AlphaFold, get the AlphaFold structure. So all of this data, the confidence data will be presented, and then you use some other tool, which could be free tool, for locating the pocket inside that particular 3D structure. Then it's like known ligand site or predicted cavity. And then you discuss the limitations, like water is not present, the flexibility is not accounted for by, cofactors are not present, the validations, all of these things you can do it. So as a teacher, as a pharmacy teacher, you should actually use AlphaFold. Now AlphaFold 3 is available, yes, to academics and to everybody, I think, recently. So you should use, maybe you can use a target structure which is experimentally known, and then the AlphaFold structure. Most of the times it is going to be very similar, but just try and see an experimental structure, say PDB structure, with the ligand and the AlphaFold structure. How do they match in terms of the ligand site, the flexibility, the induction, everything, meaning induced fit, something like that. And then based on that, you design the next experiment. Could be mutagenesis, could be docking, could be assay, or could be any other biophysical methods like say surface plasmon resonance, something like that. Then case study two, that is the generative AI from hypothesis to clinic. So this is the molecule, structure of the molecule, rentosertib, which has been discovered by in silico medicine. So this ISM stands for in silico medicine. Currently, this molecule has entered in phase 3. It has been indicated for idiopathic pulmonary fibrosis, meaning whose cause is not known. So they have, in fact, they have developed their platform, Chemistry 42, and then they also have Pandaomics and then multiple other platforms. So they claim that they have identified this particular target, that is TNIK, TRAF2 and NCK interacting protein kinase. So the AI helped them identify this target. They could relate that this target would have implication in the idiopathic pulmonary fibrosis. And then the next level, AI then helped generating novel molecule. And this one of these molecules has now entered in phase 3. And this has been done in less than like 5 years. So everything designed, developed by AI. Of course, the experimental part, they ended up doing this. So AI supported the target and molecule generation, TNIK inhibitor profile for pulmonary idiopathic fibrosis. Consider this as a case study or as an exercise. Preclinical validation and clinical evaluation required, and then a success story in progress, not proof that AI bypasses biology. So basically, what it is trying to tell is AI was used for majority of the functions, but then of course, experimental validation was needed. So as an exercise, I would request participants to ask these questions. What exactly was AI generated? What experimental evidence supports the target? What were the safety and efficacy endpoints, and what would count as real clinical success? Read about rentosertib. It's a fantastic, if you are passionate about learning about AI, this is one of the case studies, the real evidence, real life evidence, that how AI along with the experimentalists can help advance the molecules in fast track mode. This is another case study by us with this particular company named Rasayan Labs Incorporated, which is California-based company. Arshit is the Chief Technology Officer. That's me. This is my student, and Mr. Shobhan Shah is the owner of Rasayan Labs. So on October 30th October 2025, we put this paper on KM Archive, AI designed family of dual COX-2 MPGS1 inhibitors to treat inflammation with greater precision and effectiveness. So we spent 1 month. So first, of course, we developed all our tools and then used all these tools, different AI models for designing dual COX-2 and MPGS1. So MPGS1 is a target downstream of COX-2 for prostaglandin E2 synthesis. So there is a theoretical basis, literature evidence or I wouldn't say clinical evidence, but experimental evidence, biology evidence is there. So we combined this and then tried to come up with novel molecules using everything from our platform. And then currently these molecules are being synthesized and being tested. So we also filed a patent, and as you can see here, in less than 1 year's time, the downloads have been in excess of 1,000, which is itself a milestone. This is an amazing paper. If you're interested in small molecule drug discovery, you should read this paper. This is another paper that we have come up with. This was put later, I think April or May, on bioRxiv, where we say that we have designed like computational design and immunoinformatics evaluation of a multi-epitope vaccine targeting podoplanning, which is the antigen in glioblastoma multiforme. Basically, I would say the CNS cancer or brain cancer, which is very aggressive. Now here we could everything is done using AI tools. Some of these are free tools. Part of this workflow, including the 3D structure prediction, was done using the Rasayan.ai platform. Right, so this is amazing study if you are a biologics person, or if you're interested in biologics vaccines. So you can potentially read this paper. So again, the whole idea is how AI can help in terms of accelerating the timelines and doing something which was very difficult or time consuming earlier. Few slides, and I should be able to finish in next 10 minutes. And if time permits, I'll show you the demonstration, or maybe separately we can have a demo later. Okay. So, so these are the common, now, of course, now you heard me a lot about different aspects of drug discovery, but what about the pitfalls, right? So data leakage, that's another issue. Benchmark chasing. So you are trying to improve your, matrices, validation matrices, but it doesn't really add when it comes to decision. Wrong endpoint. If you use the wrong endpoint, the AI model will be good for nothing. Over optimization, the model exploits scoring loopholes. It tries to improve the statistics, but there is no real value in this optimized model. Good for nothing in the real world. Hallucination, I already talked about. No prospective test, in the sense I'm moving ahead. If the model never faces new chemistry, it's not going to improve as far as its outcome or usefulness is concerned. So the solution to this is design the validation strategy before training the model. You should be the most dangerous critic of your own work, that is your own AI model. Then only the world will if it is really good, the world will accept. Then reproducibility and the fair data. When I say fair, I'm talking about findable, accessible, interoperable, and reusable. So the AI-enabled discovery should be findable, accessible, interoperable, and reusable wherever possible. So this is another very important aspect. Otherwise, it is just some fancy stuff, good for nothing, right? So this is important. Then regulatory and ethical framing. So of course, AI outputs can support decisions, but accountability remains human and institutional. You cannot simply say that AI predicted this, it is all good, and I don't have to do experiments. No, it is your responsibility to show that whatever the AI does has actually meaning. So that's where the regulatory expectations are fit for purpose validation, transparency about data and intended use. It's like now we are using multiple models, including Ames mutagenicity model. And this is where I need to declare at the outset that this is what data I have used and this is what the intended use is. risk-based model governance. In the sense, I'm telling you, the model will do well, but then wherever it flags, if wherever there is low confidence, you need to, based on that risk, you need to use that model for whatever purpose or objective you want to use it for. Audit trails and change control. So wherever there are the things need to be changed, so these audit trails and then change control must be exercised. Ethical expectations, confidentiality and IP protection, bias and population relevance, explainability proportional to risk, and then avoid unsupported clinical keeps. I mean, otherwise, the AI will give you some fancy stuff. It may not really mean anything, so you should not chase such thing. But of course, this is where the human in the loop connection comes in picture. So FDA's recent AI documents emphasize context of use, risk, credibility, and transparency for AI used in drug development. This is very important. Now, these are the some of the free tools that I'm going to talk about. Free tools, the OCSR.ai. Please go to this website, OCSR.ai. You will be able to, all you have to do is either you can draw the structure or you can upload the structure as an image or as Smiles, name, formula, whatever CAS number, and it will generate a lot of data using the tools that we have developed together with Rasayan Labs Incorporated. So please use this tool. It's going to be fantastic. You will love it. It's all free, although some of the endpoints need to be done on the subscribed or the proprietary platform. This is just a trailer of what the platform is able to do, and you will just love it. Then this is the main platform that it's not free tool actually. The compound intelligence. So the moment you put the data, so these are some of the examples, the moment you put data, it will generate so much of data. It will also suggest retrosynthesis pathway. It will also have the formulation aspect. It will also have the toxicity endpoints. It will generate a lot of reports. So complete workflow, we have developed tools for the complete workflow. Please go to rasayan.ai and ask for trial version or demo. Right, so we will make sure we will give the demo and give you access for at least a week or two for trying out these things. And as far as academic pricing is concerned, it's very much doable. Any college can easily afford it. But before that, you have to use it and see whether it is really of any use to you, and that's very, very important. Okay. So the message is the future of AI in drug discovery is not replacing chemists or biologists or any other human for that matter. It is giving chemists a more powerful evidence integrated way to decide what to make, what to test, what to stop, and what to learn next. So it's extremely important. And with this, I thank you all for patient listening. And I'm happy if there is any question. I'll be very happy to talk about. I'll just try and come out of this. Okay.
Video on பண்ணிருங்க. So participants can interact with sir.
Sir, good afternoon, sir. So this is Dr. Senthil Kumar from RVS College of Pharmaceutical Sciences. Is there any model for ADMET, sir, in vitro or in vivo? There is a drug stability as
Pardon,
well as there is any model for. Hello, sir. Whether it is audible, sir?
Yes, you are audible.
Yeah. It is any model is available or any tool is available to predict the. Hello, sir.
Sir, just 1 minute. Just 1 minute. I got out of the meeting. Yeah, please go ahead.
Yeah. Sir, is there any model is available for DMPK, sir? In vitro and in vivo DMPK?
Yes, sir. We do have many different things, but like I said, because of the time, we couldn't really, we couldn't really do it. So basically, there are multiple things in DMPK. So in fact, it can predict the drug metabolites. It can also predict the toxicity of these metabolites. And then in addition to that, it is also able to give you multiple endpoints. So it can predict the dog. In fact, OCSR.ai, we can have rat pharmacokinetics predicted, dog pharmacokinetics, mice pharmacokinetics, human pharmacokinetics. In the main platform, there are around 728 ADMET properties related to DMPK can also be predicted. So a lot is there, sir. Please visit Rasayan.ai and get
Can, can, can you
yeah.
repeat, sir? That one? Rasayan.
Rasayan.ai. I'll just put it in the chat box.
Okay.
So Rasayan.ai. That's one, and OCSR.ai. OCSR is free. Anybody can use it for as many molecules as you want. Rasayan.ai is again proprietary. Please revisit the website and then request for demo. Just kindly share why rescoring is important, since docking score is not a good criteria to choose. Yeah, that's fantastic question, Dr. Sarvanan. Thank you so much for this question. Now, I'll just quickly answer this question. Now, when it comes to docking score, like physics-based docking, so typically around 30%, 20 to 30% hits are going to be false hits, or I would say false positives. So in that case, if you really have rescoring, so it will identify those false positives. Now, in that case, rescoring, again, rescoring, in fact, we have rescoring, which is based on quantum mechanical training model, which does extremely well to represent the electrostatics for that matter. And we have seen that with the rescoring, we were able to remove these false positives. Like some of the decoys, we purposely put it in the data for the virtual screening. And after rescoring, most of these decoys, I wouldn't say all of this, most of these decoys were actually taken out. So yeah, rescoring is extremely important, sir. In fact, if you are interested, you can reach out to Rasayan.ai, and we can have a complete, I would say, complete demonstration of the capabilities. I can spend as much time as you want. That is possible. Okay, which score is best for docking? Now, again, how rescoring strategies is utilized till now for target specificity, especially for targeting paralogs. Now, again, when it comes to now, at least I can talk about our models. So typically what we do is when we actually start, we have one layer before we actually get into the physics-based docking. So we have a layer, a deep neural network model, which we call it as deep drug protein engine, or DDP. So it's not docking, it doesn't do docking, but it tries to look at the different aspects of the query molecules which are to be virtually screened. It could be a very large collection, and at the same time, it will also look at the target binding site, and it will extract the information, and it will try to match both. And then it is like a pre-screen. For one of the targets, we have screened a large library of around 1.2 million molecules in less than 4 hours. So where we got rid of most of the false positives, or I would say the DDP engine actually gave us the enrichment. And then we only looked at the top 10,000 molecules for docking. And after that, we have used our own proprietary rescoring function, the quantum mechanics based rescoring function to represent the electrostatics or different electronic effects. And we actually came up with the top 100 molecules. And currently these molecules are being evaluated in the experimental assays.
Any other queries from the participants?
Okay, kindly share your opinion on explainable AI, which is quite popular nowadays in CAD. Yes, fantastic. In fact, we have also developed a tool. Currently, it is not available to the users, where we call it as agentic AI for drug discovery. So basically, the AI, you give a prompt, and the AI will eventually put everything in proper perspective, execute the complete workflow. So this is again an AI which is not like a general LLM. It will give you the results, and then you will be able to see everything happening in front of your eyes, and the AI will able to ask you what is it that you would want, and at the end of it, it will also explain to you whatever it has done, how it has done. We also have the explainable AI in the formulation module incorporated, where it is telling us. So if I'm looking at, say, the drug excipient compatibility at, say, under accelerated stability conditions, so it is explaining me why is this particular excipient showing this particular incompatibility or interaction. So, and then it will also suggest the formulation strategy for that. So yeah, there are multiple flavors, different names are there, but ultimately what I insist is that human in the loop connection, do not miss that human in the loop connection. Otherwise you will end up in no man's territory, and that's going to be a disaster actually.
All of you can unmute yourself and ask the questions. There is a chance that your questions in chat
Hello?
box.
Hello,
Yes.
sir.
Yes,
Sir,
yes, yes.
sir, is this OCSR limited to chemical structure drawing, or can it also used to predict other pharmacokinetic properties, sir?
No. Pharmacokinetic properties. Like I said, there are multiple pharmacokinetic properties as well. And like I said, you use OCSR. It will, and then once you have a flavor of that, you can just write to rasayan.ai, and we would be very happy. I mean, the company team, I'm just a scientific advisor to them. We'll be very happy to help you, show you the demonstration, use, meaning the capabilities. And if you think, these are useful to you, and like I said, academic pricing in nothing, any poorest of poor college will be able to afford it. So the whole purpose is for people to use these tools, and we also want to explore this. Like now, if you see academics like PCI syllabus everywhere, the AI has been incorporated. So we have to train students. So unless we as teachers do not use it, it's not really going to, we won't be able to pass it on to the students. So we have to learn first, and then I'm not saying you become a coder or this, but at least on the application side, if you can develop the thought process among the students, the responsible use of AI, that's going to be amazing. Believe me, that's our responsibility. If we fail to do our responsibility, fail to do our job, the students are going to be misdirected, not focused, and that will be more detrimental to their professional journey, professional development. There are many questions. Please write to me separately. I would be very happy to, I'm just putting in the group chat my email address. Please write to me. I will be very happy to talk to you or answer your question. I'm just putting Mumbai.edu. This is my email address. Please reach out to me. Thank you so much for all the nice comments. I'm very happy that I could contribute whatever little I know to you all. Please feel free to reach out. I would be very happy. I have visited JSS College of Pharmacy, Ooty, long ago, 2013-14. Along with a delegation from NMIMS, 3 of us, and we had a fantastic time in Ooty. I still remember all the like meeting. The facility, of course, is fantastic, and then even Ooty was amazing. Very fond memories from that trip. And Dr. Praveen, I'm actually connected. I don't know if he still works with JSS College Ooty. I'm not sure, but Dr. Praveen TK, I was, I'm in contact with him.
So Dr. T.K. Praveen is the principal of JSS College of Pharmacy, Noida right now.
Okay.
He was here in the Department of Pharmacology. He was heading the department.
Yes.
And recently, 2 years before, he got the opportunity to move there.
Perfect. Yeah, he's an amazing scientist. So we have interacted on multiple occasions. Yeah, amazing.
Right now, he's in Noida and looking after the JSS College of Pharmacy, Noida, sir.
Okay, amazing, fantastic.
So I request all the participants to reach out to the sir for any further queries or the mail ID given. Even I can share it with you.
Yes.
For the time constraint, we have to wind up the session.
Yeah.
In fact, sir has taken a very extensive session. It went up to two plus hours. And I thank every participants for the kind cooperation, keeping your name in a lengthy way, what it is appearing in the registered form. But still, I could see there are many names which are not matching with your observation registered names. And there are Lenovo, and other things. Please do avoid. So we need to cross verify things and to distribute the certificates. Hope you understand. Please do not do that. And it's time to thank Dr. Prashant Kargar. And we are very thankful to you, sir, for the accepting to be a resource person. Immediately he accepted, and in fact, no, I was just demanding him to deliver certain topics, and even for that, sir readily accepted that. The session was very elaborative. We started with detailing about data, model generation, model training, model validation, and all fundamental things are very essential for all the researchers, be it a beginner, be it a researchers, they are into the research for a long time. And even he has given much enlightenment, insightful details on the some of the research outcomes of his own laboratory through the case studies. Also, sir has given much highlight on the AlphaFold, how it can be used, and all those things. I think really the session was very useful for all the participants who are alive, live, concentrating. So we expect the same thing from the tomorrow onwards. So we don't have proper words to thank you, sir, Dr.
Thank you so
Prashant
much,
Karkar. And with a folded hand, I thank you
Thank
for
you so
your
much, sir.
extended support. Thank you very much, sir. We'll meet
Thank
in
you
some
so
other
much.
occasion. We'll make
Definitely, sir. I look forward to that. Very high regard to team JSS, who team Mysore. We have visited Mysore as well. So amazing vision that you have. I see the clinical pharmacy or hospital pharmacy that you have been nurturing for all these years. I have no words for that. Like I said, very fond memories to for Swami, like visit to the mart and then the hospital. So it's it's all blessings. I think it's the blessings of Swamiji and everybody. All the good work that team JSS is doing. I think that's unparalleled, and I'm very fortunate to have contributed to a smaller cause. And I have no words to express my how happy I am. Thank you so much.
whatever you said, all are true, sir. And it
Yes,
is all because of the Swamiji. We are here and
yes,
we are executing.
indeed,
And so even from the campus, we have a lot to do, lot more to do.
yes.
So we request your support in future also.
Anytime, sir, anytime. I am very happy. I'm just a call away, sir. You have my number. I'm just a call away, sir. Anytime, please reach out, anything that is needed. Yeah.
Thank you very much for all of
Okay.
you. And I'm extremely sorry for the initial inconveniences. We'll make sure that it won't repeat in the tomorrow session. All of you can sign off. Thank you very much.
Thank you so much. Thank you so much. Bye.
Shut up.